As we're going to be doing a DNA mini-prep, and we're going to be trying to get some plasmid out of some cells. So we have some overnight culture right here. In this case we have MC1061 cells with a PKD46 plasmid in there. So first thing we're going to do, get a 2 ml epi tube, like this. Fill it up with culture from overnight. Now, in the past I've shown you, you can just kind of grab your P1000 and take out a couple volumes and do it like that. You can also, if you're feeling lazy, probably not the best way to do it, is actually just pour it in. You need to be a little bit more careful if you're going to do it like this, because it's easy to overfill it. Just kind of pour it on in. Go almost. There we go. And I filled it almost to the brim. You don't want to go too much higher than that. Otherwise, when you cap it, you might spray some of that culture around and it gets dirty. So we have our culture. Got to label it. So this is PKD46. And we're just going to spin that down to pellet it. I only have one sample, so I'm going to get a full balance. Here's our 2 ml full balance. Make sure that it's labeled differently. And give that a buzz for 30 seconds. While that's spinning, I'm going to grab my P1 from the fridge. Oh god. And make sure my P1000 is set to 250 microliters, which it already is. Hit my tip. And now we just got to wait for our cells to spin. A few more seconds, and we're going to be good to go. Here's our PKD46 cells, and you can see we have a nice little pellet at the bottom there. The media is still a little bit cloudy. If we gave that a little bit longer, it would probably clear up a little bit better. But this is good enough. So we're just going to discard our supernatant. Again, there's our pellet still there. It's a pretty solid pellet. And we're going to resuspend in 250 microliters of our P1, our resuspension. So we get 250. Throw it on in. Eject it. Recap. This immediately goes back into the fridge. And then we're going to cap and resuspend. So I like to resuspend with that pellet facing me so I can kind of keep track of the progress of how well it's resuspending. And this one is not resuspending that well. Oh, there we go. Almost. A few more seconds. Now, if we were to be using a 1.5 ml tube like this, the pellet would have been at the bottom of that corner, and it makes it very hard for the liquid to swish around in there and break it up. Again, which is another reason why we're using this 2 ml tube. It's a lot easier to resuspend. So we're now resuspended. That's step 1. The P1 buffer needs to be kept cold again. It has some enzymes in it, and it resuspends the cells in the conditions that will allow the next couple of buffers, which are the lysis buffer and the neutralization buffer, to work their magic in order to release the plasmids and get them in a state where we can purify them. So step 1, resuspension. Step 2 is going to be lysis. We're going to crack open our cells using buffer P2, which is hiding from me somewhere in here. E, all the buffers I need except P2. There we go. And we're going to be using 250 microliters of P2. So same volume as the P1. There you go. And then at this stage, we're going to be very gently rocking it. So right now we're going to be lysing the cells. And if you shake it too vigorously, then you're going to be shearing the genomic DNA, which is going to contaminate your mini-prep. All that fragmented genomic DNA will be sticking to the column that we're eventually going to use in order to get our plasmid DNA out, so that when you run it on a gel, instead of having a nice clean band, you're going to have a huge smear with contaminated genomic DNA. So we're just going to let that rock back and forth for a few seconds and let that sit and cap our P2. And the people at Keogen were very smart, and they tried to come up with a very easy way of remembering how to do this protocol. So they have all their buffers named P1, buffer 1. P2, that's step 2, buffer 2, the lysis. And then the final one is N3, which is, step 3, the neutralization buffer. And that's what we're going to be using next. So we just get our N3, and N3 is the only buffer that we're going to be using that is not 250 microliters. You have to use 350 microliters of this one. And the way I remember it is that it is the third buffer that you're using. It's called N3, so 350 microliters of it. So we're just going to open up our tube, take 350 microliters of N3, squirt it on in, recap that, put it back up. And at this stage, you still need to be a little bit gentle with your mini prep, because you can see that after I added that N3 in, the upper half is kind of white. That's all the precipitate, that's all the proteins and genomic DNA. But the bottom half is still our lysed cells, and we have that genomic DNA in there, which if we're too vigorous at this point, we can shear it and mess it up. So we just start off by slowly and gently rocking it back and forth, kind of mix that N3 and that cell lysate up. And then we can go a little bit faster and then kind of work your way up into a good shake. And then once most of it is neutralized, it's all the genomic DNA, all the stuff we're worried about contaminating our cells with. They've been kind of crashed out already, so now we can just homogenize it a little bit better. And then this is ready to spin down. So we would spin this down for 10 minutes, which we're going to do right now. And we've added 250 of P1, 250 of P2, 350 of N3, which is about 850 microliters total. So we have an 850 microliter balance here. And you just toss that all into your centrifuge, and then set your centrifuge for 10 minutes is what you want to do. We'll do 10 minutes, and then hit start. And then we're going to end up with a tube that has a bunch of white gunk kind of pelleted on the side and on the bottom of that tube, and then some clear liquid. And the plasmid DNA is in the clear liquid, and we're going to come back at that step and finish up our plasmid DNA mini prep.